Namaste students, Kushro Vidya. I, JP and Dr. Vijay, Assistant Professor at Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the application of actin. Actin is used as a scientific and technological laboratories as a track for a molecular motor such as the myosins, and that is either the muscles tissue or outside of it, and having a necessary components for a cellular functions which can be used as a diagnostic tool in several or variants in application to specific pathogens or uh, in the nanotechnology, the myosin actin myosin systems act as a molecular motor that permits the transport of vesicles and organelles throughout the cytoplasm, which may be it may be possible for the actin could be at the, applied to the nanotechnology as a dynamic ability to have first in a number of experiments, including those carried out the acellular system. The idea behind it is the microfilament as a track to guide the molecular motors that can transport in a given load. So that the actin could be used as defined as a circuit along with the load that can be transported in a more or less controlled or a directed manner. Actin is used in internal control in Western bloating as in certain and equal amount of the protein has been loaded on each lane of the gel. In example for the left side, uh, the total protein is loaded in each well of the reacting the beta-actin antibody, which can be used as for the reference. The use of the actin as an internal control is based on the assumption that the expression of the practically constant or may depend on the experimental conditions. So it may be possible to obtain a relative quantity which can be compared with the different experiments or whenever expressed in later constants. So it may be for the desirable gene stability. For health, some alleles of the actin may cause disease, and that is the reason the technique may be help in detection of the certain uh, actin can be used here for an indirect marker in surgical pathology or some other variations pattern distribution of a tissue or a marker in inversion of the neoplasia or vasculitis or other conditions. The actin clues associated with the apparatus of the muscular contraction in the level of the skeletal muscles, which diminish of the tissue atrophy and may be used as a marker for the physiological process. In food technology also, it is possible to determine the quality of certain processed food or such as sausage by the quantifying the amount of actin present in the constituent meat. Traditionally, a method that is used based on the detection of the 3-methylhistidine or in hydrolyzed sample of the product. This compound is present in the actin and F-myosin heavy chain. So to generate this compound, a uh, flesh derived from the methylation of the histidine residue present in both the proteins. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session.